So here's our lesson on chapter 4. We're moving on past the themes of biology and into the first real topic, which is cell theory. And so we're going to study the parts of the cell today and then move on to the different processes that are necessary for life, because the cell is the smallest unit of life. And so we're a multicellular organism. It really doesn't matter whether you're looking at the cells of an elephant or the cells of a mouse, they have about the same size. So we're going to think about a cell in terms of an analogy. And so hopefully you had some time to do the pre-reading where you took a look at all the different organelle names of a eukaryotic cell. And then the last question was asking you to come up with an analogy. Now, several of the videos that are down below on Moodle that are linked here have some great analogies. So you could pick a factory, you might say a city. I think the textbook says a cell is like a castle. And so basically what they're saying is all the different components of a factory or a city or a castle are akin to the different organelles, and the different jobs that the organelles have are the different jobs of a component of a city or something like that. And so analogies can be useful because they take something that's new that you're learning and tie it to something that you understand well. And so when you come up with an analogy, it's really good to pick something that resonates with you, something that you understand really, really well. And that makes it a little bit more of a powerful analogy. Now, granted, sometimes the links between them are a little weird, but they can be your own version of weird. So I've had it also said that a cell is like a hospital. And the person conveniently said that endoplasmic reticulum was the ER of the hospital. Now, I don't know if that's like super accurate, but those kind of things. Okay, so the idea with cells is that they're going to take things in, they're going to use them, and they're going to build new molecules. So we're going to continue on what we were talking about in chapter 3 with all those macromolecules. I'll try and give them some highlight as we move through the different organelles of the cell. So these are different cells. These would be cancer cells. This middle one is Giardia. It's a waterborne illness. If you've ever done hiking or bushwork, it's the dreaded beaver fever. It's a horrible gastrointestinal waterborne parasite that you can pick up. This is Alodia, this is seaweed, and this one is probably the closest to the cells that we're going to look at under the microscope. So it's a plant cell. You can see the organelles here that photosynthesize, and plant cells are really orderly. They're always in this nice, neat grid. And so, yeah, we'll take a look at more and more cells as we go through the course. So cell theory, cells are the fundamental unit of life. They're called cells because the preliminary scientists were looking at them under their really crude microscopes, and they saw them as like a barren monk's cell, like nothing in it. Well, the whole topic of chapter 4 is all the stuff that we're going to find in a eukaryotic cell. So they weren't totally accurate, but given their limitations of their microscopes, that's what they called them, were cells, because they couldn't see anything inside them. They were barren cells. If we were to talk about cell theory, basically all living things are composed of cells. So not only is it the smallest unit of life, everything that is alive is made up of cells. And the key thing here, one of those characteristics of life, is that reproduction, and cells come from other cells. So microscopes are important in our work with cell biology because they're the way that we can see them. So we have the magnification, how big things are, and you could have times 40 times 100 times 400, and we're going to do that in our first microscope lab, whether you're doing it in person or the simulation. And then there's also a times 1000. So this is how much bigger the cells are that you can see. And so that's what increases in apparent size. That's that magnification. These are total magnifications. Resolution is the measure of clarity of the image. So, you know, it's that kind of focus, but also the limitations of the machine. So limitations are part of most tools. Light microscopes are what we're going to look at or look through in the lab. And this is a paramecium down here, one with the light microscope and one with a scanning electron microscope. Now, granted, you know, he's fuzzy and cute. we're not going to see images like this in the lab. We're going to see images like this in the lab. And honestly, this one's even a little bit better than anything I've seen in our lab with our microscope. So that's just the idea of microscopes. Light microscopes, like they sound, they use light, and that's what we use in the lab. And they're useful for studying animal and plant cells and some structures within the cells, but not all. So you'll be able to see some things like the nucleus and stuff. Electron microscope or a scanning electron microscope are not what we use in the lab. They are very powerful and they're really, you know, interesting images. You'll find them all over our textbook, all over the internet, but again, they're beyond our capabilities in the lab. You're not going to be able to see cells without the microscope. Bacteria, being prokaryotes, are smaller. That word it's later in the chapter, but it was also earlier on in an earlier chapter. That's when you need this 1,000 times magnification. Now, that's called the oil immersion lens in our microscopes. And so you need that to see bacteria, and we will use that a little bit in our lab. No, mostly eukaryotic cells, mostly plant and animals, are 10 times larger than that. And so you can see them with the 10 times, the 100 times, or the 400 times total magnification. This is low power, medium power, and high power for us. Not quite the highest, because that's the 1,000 times, but you get the idea. There is this slidey scale. It really does echo the hierarchy. You know, we talked about organizational hierarchies as a theme in biology, and this whole course is organized like that. So we've moved past the molecule stage, which is what we were doing last chapter. So we basically have, you know, chapter 2, chapter 3, then we're going to be up to chapter 4. And so, you know, this is the hierarchy, and you can zoom in. This image is from your textbook, but I'll put this slidey scale on the Moodle site because it's an interesting image of just giving a relative size of all these things that we're talking about. It's awkward in this teeny tiny world. This is the first that we're able to see anything, but we still need a microscope to see them. Now, cells are really tiny, and the key thing with that is maybe the most powerful or one of the first illustrations of that theme of form and function. And so if you're a mathy person, this slide has a whole number ratio on it. And if not, I have some other sort of analogies or ways of thinking of it that can explain how or why cells are really, really small. So if we were to take a look at cells, they have to obtain everything through their surface. So everything comes in and everything goes out through their plasma membrane, their outer boundary. And so surface area to volume is really important. Surface area to volume ratio explains why cells are small, and it is an illustration of form and function. Basically, what we're saying is there's a sweet spot of efficiency where a cell has an adequate amount of surface to serve the inside cell volume. And so the surface area of the cell is important because that's where things come in and things go out of the cell. A small cell has more surface area relative to its volume and is more efficient. And so efficiency is often at the heart of this form and function theme. So down here we have two images and they have the same volume. So, you know, there's one big blob over here and lots of big blobs over here, or lots of small blobs over here. And so if we were to take the volumes are the same, but then compare the surface area of these, you can take and divide the surface area by the volume to get a whole number ratio. If you are not a mathy person, I would suggest that if you've ever wrapped presents, if you have one big box to wrap, you're going to use an amount of wrapping paper. But if you had to wrap a whole bunch of small boxes, you're going to use more paper. And that's illustrating this larger surface area. And so basically what we're talking about here is that sweet spot of efficiency. And so cells can carry out all the functions that they need because they need to have that surface area optimized. We've said these words before, prokaryotic and eukaryotic cells. I remind you that you are a eukaryotic cell. If I were to talk about the two one more time, prokaryotic cells we talked about as being bacteria, and one of the domains being archaea. They're both prokaryotic cells. They're simpler cells. They're smaller in cells, or smaller in size. They have an outer membrane. They have a few organelles, but no real nucleus, no nucleoid, basically. Or sorry, they have a nucleoid, no true nucleus. The other thing with bacteria and archaea, they're a little more like plant cells, which you may have acquainted yourself with a little bit in the pre-reading, where they have a plasma membrane and a cell wall. And so this cell wall gives them an added measure of protection. We're going to focus more on eukaryotic cells throughout the course, but we will see some bacteria in the lab where we need that oil immersion lens to look at those prokaryotic cells. Now, on the other end of the continuum here are eukaryotic cells. Eukaryotic cells, plants, animals, whether they're single-celled or multicellular, and then also fungi are eukaryotic cells. They're basically the kinds of cells that we're going to talk about through the rest of this chapter. So they have a membrane-bound nucleus as opposed to the nucleoid, just that region with nucleic acid in it. And they have several other organelles. These organelles are the topics or maybe the terms that you defined in your pre-reading. This is what we're going to study. Now, I put a link in here. So I really enjoy thinking about evolution and links and explanations for how things came to be. And so basically we have an endosymbiotic hypothesis. And this endosymbiotic hypothesis is the link between the two kinds of cells. So basically, if prokaryotic cells are older, how did they evolve into eukaryotic cells? And the endosymbiotic hypothesis suggests that there was a prokaryotic cell hanging around one day, and it happened to eat another prokaryotic cell. And instead of digesting that prokaryotic cell, that eaten prokaryotic cell seemed to provide some benefit to the one that did the eating. And so instead of digesting it, it kept it around. And so examples of these kinds of organelles in eukaryotic cells would be the mighty mitochondria, the powerhouse of the cell, if you will, and the chloroplast. That's the organelle that carries out photosynthesis. Now, the evidence that these two organelles are more like early prokaryotic cells is that they actually have their own nucleic acids. And so they have their own DNA in them. And so this is part of tracing back human evolution back to a mitochondrial Eve. So basically, if we have an animal cell, so an animal egg cell and an animal sperm cell, the animal egg cell is much, much bigger than the animal sperm cell. And so the egg cell has basically a starter kit of all the stuff that the new organism is going to require. And included in that is the organelle of a mitochondria. So basically what this concept of the mitochondrial Eve in human evolution is saying is that you and everybody got their mitochondria and the mitochondrial DNA that came with it from their mom. And so we have this maternal lineage that we can theoretically trace back all the way to a single origin. And so that's the idea. The endosymbiotic hypothesis is just that, link between prokaryotic cells and eukaryotic cells and explains how they may have evolved. But we can, look at the evidence behind this. And I find it quite fascinating to think about. So we're going to get into the different organelles of the cell, and your textbook divides the functions of a cell into 4 major categories. I'm going to take and divide that up a little bit smaller, a little bit more finely, so we can look at a few things more specifically. And then I've put this in the PowerPoint. I know that you can't read it, it's too small, but this is a table from your textbook, and it's a really great chart that has the different organelles listed and their jobs or what they do in the cell. So we've got structure and function in this, sort of in a nice, neat table with fewer words than what I'm going to say as I'm trying to explain all of this for you. So the way I'm going to sort of divide this up is, like I said, a little bit more finely. We're going to talk about these different processes as we go through the cell and the different organelles that are linked to these processes. So whether you learn it from the chart, whether you learn it through this, you have your options. I would also say that being a specialist in all of the organelles of the cell is probably unrealistic. And so by picking your favorites, I think that that's a great way to make it a little bit more accessible. So we're going to start at the center of the cell with sort of the control center. If you're talking about the organelle as a castle, maybe this is where the king or the queen lives. So the nucleus is that control center. It has the genetic information in it. Now, it is also bound by a nuclear envelope. So this is a membrane. And I will remind you, for the first time, that all membranes are made up of an important macromolecule that we studied in chapter 3. They're all made up of phospholipids. And so those were one of the lipids that we talked about at the end of the chapter 3 material, and they are everywhere. So phospholipids are in every membrane of every organelle. So everywhere you see the boundaries, the membranes, those are phospholipid bilayers. And you can look back to chapter 3 if you need to refresh your memory on that, but we're going to talk about it lots in chapter 4 and 5 as we keep going through. So the nuclear membrane has openings in it. And so this nuclear membrane, nuclear envelope, either one that you want to call it, basically has these holes in it so that large molecules can pass through. We want information to come out. We want nucleic acids to come out. So the fancy word, or maybe the more general term for genetic material, is chromatin. So this is a very general term. Sometimes it's handy to know, a more specific term, but if you kind of zoom out a little bit and use a general term, you're not wrong. And so it's kind of interesting to know that vocabulary and how it nests together, because that can be really useful as you're trying to not use the wrong word, if you're following me. Okay, so we have the nucleus, the nuclear membrane, and the general term for genetic material, which is chromatin. Inside the nucleus, we also have a darker region, and under our microscopes, you are able to see these. It is the nucleolus. And so the nucleolus is a denser region in the nucleus, and in there is a different kind of nucleic acid. So we have DNA out here, that's the purple noodle-looking stuff. And then in here, we'd have RNA. You remember those were the two different kinds of nucleic acids that we talked about in chapter 3. Now this nucleolus has the information for making ribosomes. We're going to talk more about ribosomes when we get to the endoplasmic reticulum, specifically the rough endoplasmic reticulum, because this is really, that link between genetic material and proteins. So let's carry on with this. The next image is stuff from your textbook, and really it kind of just shows, under a microscope that's beyond the power of the ones that we can see. And then over here, the schematic from your textbook, showing it just a little bit more in 3D. So we have the nucleus, the nuclear envelope, or the nuclear membrane. Nuclear membrane. We have the nucleolus in there, that general term for genetic material or chromatin, and then the openings in that nuclear membrane to allow large things to come out. So what that pore is hooked up to is the endoplasmic reticulum. Those are really long words. I generally shorten it to the ER. So as we carry on with the endoplasmic reticulum, you basically have two kinds of endoplasmic reticulum. You have the rough endoplasmic reticulum, and you could think of rough or ribosomes for that R Either one would work. I link them in my mind, even though the R there technically stands for rough. And then the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum and the rough endoplasmic reticulum have different jobs, and those jobs are linked to the macromolecules that we talked about last day. It says this on the next slide, but the smooth endoplasmic reticulum is key for making more lipids. And the rough endoplasmic reticulum, those ribosomes, are where proteins are made. And so those two macromolecules were important when we did chapter 3, and we're going to carry on linking them into the functions of the cell. So here we have, those are those key molecules that are manufactured, they're altered and distributed. So I'm going to start at the bottom, number 4 down here, the rough endoplasmic reticulum. This is the protein manufacturing. The smooth endoplasmic reticulum is all about lipids. Now lipids could be more phospholipids or more membranes, because we know all membranes are made of phospholipids, and phospholipids were one of the kinds of lipids. Now, different cells in your body have different kind of specialties, and in the textbook it does say that liver cells have a lot of smooth endoplasmic reticulum because they're key in the function of detoxification. So it helps in that process, but generally, they do make more lipids in all cells of your body. So the next one, if we were to talk about this molecule manufacturing, altering, and distribution, beyond the endoplasmic reticulums, we could talk about the Golgi apparatus or a Golgi body. So generally the whole thing is called the Golgi apparatus, and one lobe is the Golgi body. In here, what happens is they take the proteins that come from the ribosomes and basically package them up. So they are the packaging plant. Maybe they're the mail room in the castle. I don't know what your analogy is going to be for these ones. So basically take those proteins and they're going to package them up. They're going to alter them. We know that shape is everything when it comes to the function of proteins, and they need to be just right in order for them to do their job in the cell. So if we were to take a look at this Golgi apparatus, and then one side, or one single Golgi body, that's like the whole kind of lobe, one piece of the bread in the stack here. Proteins are going to go in one side, they're going to get processed, and then they're going to be packaged into vesicles. So vesicles are membrane-bound little packages that can ship molecules anywhere in the cell. And so here we have vesicles coming off. And these proteins, remember that's the key job of them, these proteins are perfectly packaged for going to where they need to do the job that they need to do. All right, today is a sad day because the mitochondria, the mighty mitochondria, the powerhouse of the cell, which is what you may have thought of it as prior to this, is basically, we're going to go into more detail in this, a lot more detail as we move into chapter 6. Basically, what happens in the mitochondria is cellular respiration. So yes, it's the powerhouse. Maybe it's the furnace in your house, whatever your analogy is. But cellular respiration is really what happens when we have glucose being converted into ATP. This is the energy currency in the cell. So we'll talk more about ATP and what that molecule is in the coming chapters. But basically what happens in cellular respiration, it was an example of a chemical reaction. Oxygen and glucose are going to go in, and ATP, carbon dioxide, and water will exit. And like I said, it's all of the topic of chapter 6. And I might remind you that mitochondria were part of that endosymbiotic hypothesis, and they are a little different than most of the organelles. They have not a single phospholipid bilayer, but a double membrane, a double phospholipid bilayer. Oh my gosh. So if you were to cut through, you would actually have 4 layers of phospholipids. Each one of these is a phospholipid bilayer, because that was the arrangement with the hydrophilic heads and the hydrophobic tails. of those macromolecules being phospholipids. And so they're really important, that phospholipid bilayer. And the key thing with these mitochondria is that they may have been similar to the early prokaryotic cells and part of that endosymbiotic hypothesis. So they do contain their own nucleic acids, and it's really this membrane space is really the location that we're going to talk about when it comes to cellular respiration. Moving through, almost done. We've got the idea of recycling. So maybe it's like the recycling center or the dump or the compost pile or whatever in your analogy. So we have vesicles, which we've talked about before, as the Golgi apparatus, the Golgi body shipping proteins in. But we also have membrane-bound vacuoles. And on this diagram, there is not a lot of a difference between the brown blobby bits. And so these are just artist renderings. They're really just all little blobby bits. But the key thing here is that we can have vesicles for transport. They're like vehicles. I know that they're not, but that's the way I remember it. And I store my vacuum in the closet. So vacuum. To keep those two words that are really similar apart in my head, this is my memory device. So vacuoles are membrane-bound sacs of material. You may have answered that plant cells have a large central vacuole because they're going to manufacture a lot of carbohydrates through the process of photosynthesis. And so, yeah, those are the two. There's a specific kind of vesicle called the lysosome. And the key thing there is that it contains enzymes. So they basically are where macromolecules can be broken down into monomers to recycle those really valuable monomers. If you remember enzymes, skill testing question, what kind of macromolecule are most enzymes? Proteins. Remember that's the question or the answer when you're half asleep and I ask a question. And so, yeah, that's probably also full disclosure. I don't usually do this in the videos. That's my favorite organelle. I don't even know why, but it is. Okay, so these are that kind of image again from your textbook. So we have transport vesicles, storage vacuoles, and then the special enzyme containing lysosomes. And so those are kind of all linked. They're all on the same page here. The idea that, you have a vesicle, so these might be neurotransmitters coming out of a neuron. And so that's the kind of protein that might be or kind of molecule that might be exported from the cell or transported out of the cell. So the cytoskeleton, we have a bit of a framework going on here. We have some specificity of these. Don't get too worried about them because there are a lot of squiggly bits, if you will. We have centrioles, which I think look a lot like churros if you've ever had deep fried churros. And basically centrioles are one of those things that you might have listed as present in animal cells, but not in plant cells. We're going to talk about them in chapter 8 when we talk about cell division. And so those will come back to us as we carry on in our studies. The other thing is we have microtubules. Now, microtubules are for movement. And so we've seen that image of the paramecium. Remember the fuzzy little guy? So the fuzziness on the paramecium are cilia. You have cilia in cells of your body as well, in your respiratory system. And cilia basically, allow things to move. The other kind of microtubule might be what you might think of as the human sperm cell. So that tail, that is a flagella, and cilia and flagella are made up of microtubules, and they're part of the cytoskeleton. And basically, this one is a very nondescript thing here. Filaments, you also have filaments that basically are that structure and support in a cell. Not a lot of detail on that. Don't worry about it. So here, this one just have better images than my little weird sketches. So there's that fuzzy paramecium and the human sperm cell. So the outer boundary and the inner background material, we're almost there. We have the plasma membrane. if you remember, it's made of a phospholipid bilayer. Just to give plug to all those macromolecules that I can. The key job of this is that it controls what goes in and out of the cell. And that's what we are talking about with that efficiency, the form and function of surface area to volume ratio. The other thing that the plasma membrane has is identifiers. And so the easiest one I can come up with this is blood type. Now you have other identifiers that allow your body to understand what kind of a cell and what cell is you and what cell is not you. But if you have type A blood, that means you have A markers. If you have type B blood, that means you have B markers on your blood cells. And so that's a good identifier of cells and a good example that you might already know a little bit about. And generally, that plasma membrane, sometimes called the cell membrane, even though there's many cell membranes, the plasma membrane protects the contents of the cell. Inside the cytoplasm, it's the water-based solution that basically contains all of these dissolved items. So these are all macromolecules. They all have functional groups that make them soluble, if you remember that from chapter 3. And really, this is the aqueous environment that is on one side, the water-based environment that's on one side of that phospholipid bilayer. The last slide here gives us a summary of the difference between plant cells and animal cells. Generally in your pre-reading, you're pretty good at summarizing those differences. We won't go through all of them, but when we do get to our botany chapters, we'll go back to these. And also when we get to chapter 7, we'll talk more about those photosynthesizing things, those chloroplasts. The other thing to mention, because we will look at some plant cells in the lab, they have a cell wall and then a plasma membrane inside of that cell wall. So they have both. They still have mitochondria. Plant cells still undergo cellular respiration, but they get both cellular respiration and photosynthesis. They also, one more thing, that large central vacuole right there. And then the other thing is that they lack centrioles. They still have the same function or the same steps of function in cell division, but they lack those churros, if you will. And the last slide can help you label in your reviews. We did not cover peroxisomes, so you don't need to worry about that. And you'll find this in the review. Typically, not in our course, but in other courses, when I give a diagram of a cell, I leave these lines off because, these artist renderings of the different blobs are kind of awkward. Like, as an example, what was a peroxisome and a lysosome, how would you know the difference between those two? Other than that's where the blank is, and you would just memorize where it goes. And so, be kind with these artist renderings, the blobby bits, and especially they don't photocopy and color very well. So that's our chapter 4. We're going to move on with cell functions in the next coming chapters.